I request Nikhila ma'am to escort them. Today's speaker for this session is Dr. Bhupender Singh sir. He is the principal, secretary officer, Central Research Facility, IIT Delhi. Sir is a researcher in the field of nanomaterials and material characterization techniques. His work primarily focuses on synthesis, photophysics, and optoelectric properties of various nanostructures, with a particular interest in RNA templated nanomaterials and their applications in fluorescence, sensing, and photocatalysis. Sir, research has explored the synthesis of innovative nanostructures such as lead nanomaterials with application in fluorescence, sensing, photocatalysis, and optical properties. Through advanced characterization techniques, Sir investigated the structural and optoelectronic properties of nanomaterials, including their fluorescence, emission, morphology, and electrical conductivity. Sir has also contributed to the development of sensing platforms for detection of hazardous contaminants like trinitrotoluene and organic dyes using nanomaterials such as gold nanoparticles, carbon dots, and metal organic frameworks. Sir has made significant contributions to the field of nanomaterials and over 12 impactful publications in the high-impact journals. Sir research spans across material synthesis, characterization, and environmental applications, and he has actively mentored PhD and MD students in advancing these technologies. His expertise in nanomaterial characterization techniques has played a crucial role in developing novel sensing platforms for environmental and industrial applications. Now I request Dr. V.V. Ramareddy sir to welcome our today's speaker, Dr. Bhupender Singh sir, with the floral greeting. Welcome, sir. Thank you, sir.
Our chairperson, Dr. Vedantam Giridhar sir, has 13 years of PG and UG teaching experience and 7.5 years of research experience. Sir has published 16 articles and 11 books. Sir is investigator for 12 research projects. Sir is a member Bureau of Indian Standards Ayush Committee, member National Council for Indian System of Medicine Ayurveda Curriculum Committee, member of Institutional Ethics Committee of ICMR National Institute of Traditional Medicine, Belgavi, member Faculty of Science, member R&D Cell, member of BOS, Kaher Deemed University, Belgavi, worked as Senior Research Fellow in CCRS unit, contributed in 4 volumes of research database on Ayurveda, Siddha, other traditional medicine, and related science published by CCRS. Contributed in 5 e-books published by CCRS. I request Dr. Soumya SB, Vice Principal, and Dr. Nikhila, HOD, Department of Sushrutha, to welcome to Dr. Vedantam Giridhar sir with a floral greeting.
Welcome, sir. Now I request our today's speaker, Dr. Bhupender Singh sir, to speak on the topic Mass Spectrometry and its application in degenerative disorders. Over to you, sir.
Thank you very much for the nice introduction and felicitation, and in giving me the opportunity to interact with the audience. So I would like to this I have to like.
You can change in that, sir.
Okay. Thank you. So today I would like to discuss about the usefulness of mass spectrometry in the medical field, and as the workshop is on the degenerative diseases, so I have taken some case studies of the degenerative diseases. So this is the overview of my talk. First of all, I would like to discuss about the basics of mass spectrometry, what is the use of that mass spectrometry and various types of mass spectrometry, and next is the advantage and limitation of these techniques. So what is mass spectrometry and how it is useful? For any sample to be analyzed with the help of mass spectrometry, first we need to ionize that sample. And there are various kinds of sample which we come across in our daily life and for different research purpose. So what is mass spectroscopy? What is nominal and exact mass spectroscopy? It is just simply a weighing balance to weigh the charged particles, charged moieties. In order to, if you want to weigh some charged moieties, then you can make use of mass spectrometry. This is simply a machine used to weigh the molecules on a molecular scales. So if we need to know the m by z, mass to charge ratio of the molecule, first we need to ionize the sample in the form of cation or anion, then we can do the recording in the mass spectrometry. So if it is this heavy structure, it is having molecular formula 452, then and mass is unit positive charge, then we can do the analysis by using these formulas. One is, I want to explain you, one is nominal mass and one is average mass. Nominal mass is simply whole number, integer. Say hydrogen is having 1, oxygen is having 16, carbon is having 12. And average is the how we are having two isotopes of carbon, one is carbon 12 and one is carbon 13. Depending upon their abundance, we need to take the average. And by doing what we do, we record the mass of one isotope, that is 12, and multiply by its percentage plus mass of the second isotope multiplied by its abundance or percentage divided by 100, then we can calculate the average mass. So these are the average mass of some of the isotopes. Next, this is the periodic table. All these are given. These are the, we generally talk about the nominal mass and whole number, but actual mass is given in the periodic table. It is in the decimal places. And how they have been calculated? All of them have been calculated using mass spectroscopy or taking a reference of carbon 12. How much heavier it is with respect to that carbon 12? 1/12 of carbon 12. So these are the nominal masses of different element, and these are the exact masses up to 3 to 4 decimal places. So this carbon monoxide is having 28, we generally talk about exact mass is 27.9949. And similarly you can see the other molecules. So average monoisotopic mass, this is the isotopes of carbon, and average mass is this one. So, but it depends upon the instrument which we are making use of to ionize to know the mass of that moiety or analyte of interest. So we are having these peaks for different analytes. This is the average mass is actually this one 47.2, but it exists in the two form. One form is 4.7.3. It is most abundant. It is having almost 100% intensity. And second is having low intensity. When we multiplied this peak with respect to its abundance and second peak with respect to its abundance and divide by 100, then we will get the average mass. So this different systems are there to ionize the sample. We need to fed the sample inside the machine, then it will be get analyzed, and then analyzer will filter these moieties and mass will be detected with the help of this detector. So these are the different mass depending if your sample is in the gaseous state, or you say your sample is volatile in nature, then we can make use of gas chromatography as an inlet in that case. And when gas chromatography will be there, then gas chromatography can only separate the moieties. But when we hyphen it, couple it with the help of mass spectrometry, then it can calculate the mass of that. So this is the main requirement why we need to couple with mass spectrometry. GC cannot distinguish between the two identity which have same retention time with the column. In order to differentiate these two moieties, we need to couple it with the mass spectrometer. But if the sample is liquid, then in that case we need to couple the liquid inlet system with the mass spectrometer, and that system will be LCMS. So what are the different sources? Our sample can be solid, liquid or gas depending upon the polarity of the analyte and molecular weight and sample complexity. We can make use of different ionization systems. So what we do, one is their sample introduction. That depending upon the nature of the sample of interest, whether it is solid, liquid or gas, we can use different introduction system. Then ion source will ionize it, and after that there will be analyzer, and after that it is detected by the detector. These are the different inlet systems. So electrospray ionization, in this case electrospray ionization, it is best for the polar molecules. If the molecules you are of interest, which you are want to know the mass, then depending upon the polarity, if the sample is polar, then this is the best technique for the inlet to couple with the mass spectrometer. So these are we can make use of, depending if the sample is gaseous, GC can be used, but the limitation is this, it can detect mass, this is 100, this is 1000, we can go up to almost 700 mass. And another is atmospheric chemical ionization, and next is ESI. The range is different depending upon the nature as well as polarity, and second is the mass. Ionization, this one I have told sample inlet, second is ionization. So ionization source, it will create ions and dissolve the ion before they enter into the mass spectrometry or analyzer. So this is inlet system is not the part of mass only. Main source is ionization, second and analyzer, and third is detector. So what we do in case of ESI, ion formation, a sample is injected and it will be polar, there will be formation of drop, and with the time when we do the heatings and liquid will be evaporated, and there will be positive charge inside a small drop. These positive charge will repel with each other, and there will be formation of singly charged ion. And these after that there is vacuum. On the basis of the pressure difference, these ions will be aspirated inside the next chamber. That is the analyzer. So this is the different types of mechanism I have shown here. This is the evaporation, 10 to 20 nanometer. Then when there will be evaporation, there will be ion crowding, and then it will try to reduce its surface, and finally it will convert into unicharged. species. Charged residue models, positive charge is on the surface of solvent droplet. Evaporation continues increasing the proximity of the analyte and the charge until single charged residue remains. So it acts in two modes, positive mode or negative mode. If the sample, if we are making use of acid, acid will give H ion, and H ion if it associated, then plus H peak will come. And if base, we are having basing medium, it will take H ion from that and we will have a negative signal. So we get peak in the form of what is the molecular mass plus 1, that of hydrogen. In this case of positive mode. So these are the various species we generally make use of for ESI, MS, peptides, proteins, smaller polar compound drugs and their metabolites, and other pesticide, pollutant, dye molecules, organometallics, and negative. Some proteins are negative in nature depending upon the number of the acidic and basic group present. Since proteins are the mixture of amino acids, they combined by peptide bond, and if the number of acidic group are more, then it will be positive, and if the number of basic group is there, then it will be negative. And if they are equal acidic, then it is neutral. Then atmospheric chemical ionization, it is done inside, outside the chamber, and at atmospheric pressure, we make incident some chemical species to make it, convert it into cation, that then it is forwarded towards the analyzer after formation of ion, analyzer and then detector. So another is atmospheric chemical ionization. This is shown, there is a corona tip, and at the surface of which ionization takes place, and after that vacuum, and it goes inside the analyzer and then detector. So this is MALDI technique. MALDI is one is soft ionization and other is hard ionization. If we talk about GCMS, in case of GC the energy is higher and the species are actual molecule will be fragmented into smaller parts, and this is soft ionization. In this case we get the mass of molecule, intact mass of the molecule, and we will not get the fragments. This is best for knowing because if we are having a mixture of proteins, then and it undergo fragmentation, then large of peaks will be there. In that cases it is technique is fine as we get the molecular ion peak intact. So these are the nominal. In this case resolution is poor. If we talk about the elements, then ICP-MS is there for inorganic. If someone was talking about the arsenic and other, so there are possibility of accumulation of arsenic, and there are problem of cancer and other disease. So in this case I say that comes under the category of quadrupole and normal. ICP-MS, inductively coupled plasma mass spectrometry. That is quadrupole. In that case there are 4 rods, 2 are parallel to each other, 2 this way and 2 opposite side. They are having opposite charge, and when we apply voltage, we select an element, say we select arsenic 75 is the mass, automatically according to the mass, radio frequency to the DC voltage will vary, and only that ion will be allowed to pass to the detector. So this is accurate. Up to 3 to 4 decimal places we can make use of this TOF, time of flight, and orbit trap. This is the latest one, orbit trap. And up to 4 decimal places we can know the molecular mass. Quadrupole base, these are the 4 rods. These 2 are having positive, the 2 are having negative charge. When we switch, this polarity keep on changing, and when we apply select any analyte, only the voltage, it will resonate corresponding to that mass, and only that ion will be allowed to pass to the detector. So this is the, if we are having 310, these are the species present inside, and if you apply voltage according to 3010, select one analyte, only that will go one time. Then we change the voltage, then second will, depending upon which analyte we select, only that will be allowed, and accordingly voltage will be set between the rods. So in this way only one analyte is going 360 if you apply voltage. Second, ion guide, this act as ion guide. This ion move in spiral way and reaches through the detector and detected by the detector. Ion guide. So tandem mass spectrometry, in this case, say GCMS, LCMS, these are not sufficient for better results. We get some matrix result. False peak we can get in case of GCMS and LCMS. In order to get the only real peak, we need tandem mass spectrometry. That is, we are having triple quadrupole. One quadrupole is this one, second one is this one, the third one is this one. This will act as a filter. And second one, in case of single quadrupole LCMS or GCMS, only these two are not functional. These are not the part of the machine. So only one will act and it will give. But if we talk about iron, iron has an atomic weight of 56. But in this instrument we make use of argon gas that is having molecular mass of 40, and oxygen combined with that it becomes 56. So we will get the peak of false peak because of iron oxide with iron peak. So in order to remove that false peak, we need triple quadrupole. In this second middle part, we pass some helium gas, it will bombard the false nuclei and drop into argon and oxygen. So we will get only peak of iron, rest will be removed. So single quadrupole mein we can do single ion monitoring, one single quadrupole. There are two type of scanning. One is simultaneous and another is sequential. So in case of ICP-MS or single quadrupole, most of the are working as simultaneously, and rest if simultaneous it will be fast but resolution will not be good. But if it is working sequentially, it will take time but it will work fast. Simultaneous and sequential. So full scan means full range. Single ion monitoring, if we select one ion, and MS product ion. In this case we will get precursor ion, parent ion peak. That will fragment into daughters, and neutral ion will not be detected by the detector. So it is called multiple reaction monitoring, MRM. Time of flight. So generally I have discussed in the beginning that on the basis of mass to charge ratio we do this analysis. So it is the Q into E, electric field applied, Q is the charge, is equal to half mv square kinetic energy. Then we shifted the terms from left to right, m by q will be equal to 2EBQ. So voltage will be replaced by length upon, velocity will be changed by length upon time. And then we shift the third one, the formula we get from this 2E by L2 square is constant. There is a tube there is a tube, and analyte is have to move the path. The lighter ion that is which is having low atomic weight, that will reach the detector first and detected first, and the moiety which is having heavier mass, that will reach the detector later and be detected later. So times of flight depending upon the time taken by the analyte or the mass of the analyte, we can make use of this. Smaller will be detected first and heavier will be detected later. So I have shown this. Thus there is the different sizes. So this is the retention time. First it will reach the detector and give its signature, then it's heavier, then it is no more bulkier. So depending upon their mass, the different timing will be there. This is the retention time. How long it takes to reach the detector. Smaller will cover the rays first. So next is resolution. I want to, what is the beauty of this mass spectrometry? We need to know resolution. Resolution to see too closely point separate. This is the resolution. and it is equal to mass. This is the peak, and divided by the two neighboring peaks difference. It is 0.5 is the difference. If then it will come out to be the 630. So if the difference between the two peaks, how much is the difference? That depends upon the resolution, whether the instrument is able to remove, analyze them separately or not. So this is one peak, and it is having so many multiple peaks. This peak from here to here, we will not be able to separate these peaks. If the instrument is having better resolution, then individual peak will be resolved. It will look like this, if the resolution of the instrument is better. So if the instrument resolution is less, there are multiple peaks which will not be able to see. And if we increase the resolution, then we will be able to see them. And at resolution of 1000, we will not be able to see all these peaks. But if the resolution is 10,000 of instrument, then we can to see them separately. This is the mass and this is the resolution formula again. This is the difference between the two peaks and at which peak we are taking into picture. So difference divided, so it is, if the instrument resolution is less than this, then we will not be able to treat these two peaks simultaneously. If the resolution of the instrument is less than this value, then we will not be able to see these values separately. So this is true mass, measured mass, this is the difference, this is the neighboring peaks. So difference is 0.004 divided by, we are talking about the actual, so it is come out to be, this is the ppm. तो ये आपका 10 लाख से मल्टीप्लाई करेंगे तो आपका ये 1 ppm आएगा. 1 into 10 raised to power 6, that is 1 ppm. So this is the another system, and in this case detector, technology advancement is there. Company are busy in miniaturization of the technology and advancement depending on the sensitivity, detection limit and other. So in this case, this is triple quadrupole system. In this case, I want to show this is the extended TOF. This is the time of flight. We have increased the length of the tube. If we keep it direct, then the length of the instrument will be too much, but we have given it a W-like shape and then resolution will be better. So quadrupole, single quadrupole or triple quadrupole, this is the difference. Single case में आपका ये तीनों मैंने जैसे उसको I have given the example of iron and argon oxide. If they are having the same acidos, both of them will go outside the quadrupole and they will be reached to the detector, and it will be contribute a false peak. But in this case, the false peak will be removed by the collision cell, then it will be split the false peak and only original signal will be received in the detector. So data dependent surveys, so serial analysis, discontinuous analysis, surveys can, I want to do qualitative analysis, quantitative analysis, semi-quantitative. If you are having reference or standard, then you can do the quantification. If you are in Ayurveda, what is the main problem? We are not having standards. So how we can do the quantification? So we need standard if you want to do the quantifications. And if we are having standards reference, so we will prepare 3 different solution and record the signal, and after then we run the unknown sample, we can do the quantification. But in this case, if the standard is not there, then it will be difficult to quantify. We can only do some qualitative analysis. Qualitative will let us know the how many moieties or entities or analyte are there in the sample. So continuous, parallel, due to post-analysis reconstruction, we can do the relationship. If there is fragmentation in case of MALDI, I told that there is no fragmentation and we get the peak of molecule. That is called molecular ion peak. And if there is fragmentation, then we need to see the fragments and correlate. And nowadays, what is the best part? With the instrument we generally used to have database and library, we can match with that. These are the just data to share. These are the different peaks, and we can do the quantification. This is the mass range. This is M by Z value, and this is the intensity. The most intense peak, that is called 100%. That is called most stable or most abundant peak, and that is called base peak. Other peaks are in comparison to that 100. We take this as 100%, and rest are taken is in that reference. How much is the intensity, we take the percentage. Liquid phase separation. So this is the liquid phase LCMS. Yeah, ultra UPLC HPLC हाई परफॉर्मेंस, ये अल्ट्रा फास्ट परफॉर्मिंग HPLC होता है, और इसके अंदर कई सारे मास हम असेंबल कर देते हैं, तो ये डिफरेंट उसमें पीक्स. अब एक्स्ट्रा पीक आई हुई थी तो इसमें सिर्फ सिंगल पीक ही आएगी. अगर हम मास अगर कई लगा MS कई कपल कर देंगे, सिंगल क्वाड्रुपोल ओनली वन मास, 2 quadrupole and 1 collision cell triple quadrupole. In this ME there are too many MSMN system, and we get rest of the signal will be discarded and we get single peak. So ion mobility depends upon we give a frequency to a particular ion and only that will be detected. But there are two problems in this. If we say take a piece of paper, we crumble it and let it fall down. And we left the paper, simply let it fall. It is having large surface area, it will fall at later and it will take more time, and the paper which when we crumble it, then it will take less time. So this, if the species are having same molecular mass but different conformation, if we talk about proteins, different helical structure, alpha, beta, and on the basis of configuration, we can separate with the help of ion mobility mass spectrometer. So this is the 3D structure. ये retention time depending upon the interaction between the column and analyte, and second M by Z mass. ये dt इसमें time कितना लेता है, how much time it takes. Just I have shown the ion mobility CCS. Retention time is not sufficient for the separation of peaks. Two species may have the same retention time, but that will not give the sufficient information. That's why we need to couple with the other instrument, say ion mobility or other mass spectrometer, and in order to get the full peak. So this is, I have taken an example, this is Waldo. If you want to search this one from this crowd, then what will we are not able to distinguish. And ultra fast performing, some of them screening will be done, then the crowd will be somewhat, result will be this much. Earlier the sample was too much crowded, this is our sample and we want to search this. So it is difficult to directly find from the crowd. So there are different techniques to filter them. So one is ultra five we will get only retention time. On the basis of the retention time, same maybe having same retention, but we will not be able to distinguish. Then we other collision cell will be used. Then mass we have applied. Then we have sorted out them. And if we couple with other ion mobility, on the basis of conformation, we can separate them. And after that we may conclude, depending upon the fragments from crowd, we have reached to this conclusion. So reduce the number of false positive peaks, additional dimension of separation to obtain cleaner spectra in composite sample, improve the reliability and assignment of isomers. So this is the flow chart I have already discussed, qualitative analysis, quantitative analysis, and one will simultaneous. If it is going, it will take less time, but resolution will be poor, and if it is simultaneous sequential, then it will take time. Targeted, we need to reach the target, then we have to use the suitable techniques. So current research in applications, and so we are having some use of mass spectrometry in medical sciences. So these are some methodology I have followed. So what are the instruction if we want to use it in medical? First part is we need to take which type of mass spectrometry is suitable for our work. We want to do some, sorry. So you must have some idea about the mass spectrometer which will work good for us. So these are the ionization technique, and then there will be detection technique. Depending upon the detection limit and capability, we can make use of different mass spectrometers. So these are the simple tissues. We can take blood, serum for the sample analysis. What are the proteins responsible for a particular disease? If we want to monitor some patient, what we need to do? We need to compare it with some healthy person and monitor with the time that what kind of food he is taking and what are the proteins, extra proteins is there or not. These are called biomarkers. If you want to see some disease, then there will be some causative organism, some species which are responsible for that disease. So we can take the sample of blood, serum and other body fluids that can be analyzed with the help of mass spectrometry. With the help of this, if we are having two persons, both are having same diet and staying in the same environment, and if their blood sample, then there might be accumulation of more accumulation of one type of species or proteins, and on the basis of that we can see that this type of extra species is in his body. We need to monitor him and avoid such kind of diets which are increasing its number. So in that case we can monitor the health and by taking the different samples. So software data will help us. We are having result. Then how to interpret? There are libraries with the system which can distinguish what are the sample and what are the proteins, metabolites, proteins. If you are to have higher mass, proteins are the polymer of amino acids, then we generally make use of MALDI-TOF in order to get the intact peak. From intact peak we can get the idea of that molecular or protein. And if you are having small moieties, then you can use of LC, MS. So managing the large database, ensuring data accuracy, reproducibility. So definitely biomarker, I have told you what are the improve and importance in diseases, traditional methods versus. If we talk about traditional, they are time taking, not accurate, there might be so much errors. In order to improve the quality of the results and pinpoint the problems, sorry. So these are the traditional methods, and now we go to, these are the biomarkers. I have taken the Alzheimer's disease, and these are the cerebrospinal fluid, blood, these are kind of different biomarkers. These are amyloid, beta proteins, peptides, tau proteins, these are different. These are the biomarkers for the disease. And we can make use of this technique LCMS, MALDI-MS, LCMS, LCMSM. Then in the similar way we can study the Parkinson disease by taking their body fluids and analyzing the different functions, metabolites or proteins in the sample. Similarly, Huntington disease and others. They can be analyzed, and these are the techniques, what kind of sample we can do, what will be the result, and how can we, some of the samples we can do brain sample of tissues and all that, from that we can have an idea. Another one more disease, amyotrophic lateral sclerosis, and this multiple sclerosis, this is body fluid, these techniques can be analyzed using. And this is another, nowadays this is the latest technique I would like to, multi-MS imaging. Multi is the inlet, mass spectrometry imaging. With the help of this, you can see the, you take a, cut a section of the tissue. If you are working on mice, then you can cut a section and insert in the machine, then you will get the location as well as proteins or biomarker which is responsible for that disease. So this is the mass spectrometry imaging. So this belongs to 888 peak. This is the protein having mass, this one, and above is the protein having this, and this is their respective intensity, which 100% is red one, and there is a gradient in color. So with the change in color, the concentration is carrying. So we can pinpoint the position, location, as well as the proteins distribution. We can have the mass spectro intensity versus M/Z ratio. And these are the multi-MS protein from rat kidney. So these are the masses. Different masses I have, and different colors have been given. So from that we can match the library and can know about the, which are the responsible proteins or nucleotide, which one is responsible for that. So future personalized MS can facilitate the development of personalized treatment plans based on individual proteomics and metabolomics profiles. Non-invasive MS is mass spectrometry advancing towards blood-based or urine-based diagnostics tests for degenerative diseases and therapeutic targets. MS is instrument in discovering new drug targets. So if you come to know that this is the proteins or this is the multi-responsible, you can try it that whether I can give this drug or it will control. After that you can take the sample and analyze there is some control or not. Thank you. That's all from my side.
Thank you, sir. Now I request our chairperson, Dr. Giridhar Vedantam sir, to give up the remarks for this session. Any questions?
House is open for the questions. Anyone want to know something from my side?
Any questions from the delegates you can ask, please.
Namaste, sir. Thank you so much. Sir, with a limited physics background, I tried to understand, and I have, my question might sound a bit silly to you, sorry for that. In Ayurvedic medicines, we use heavy metals a lot, and before using them, we process them a lot, and we believe that the nature of those heavy metals have changed totally, and they have garnered the medicinal value. And there are thousands of heavy metal compounds in Ayurveda. On the other hand, what herbs we are using, those are also having lots of heavy metal contaminants. So when I am preparing a medicine for my patients, I might be using purified heavy metals according to our Shastras, and my herbs which I am putting in that, they might be having contaminants of heavy metal. So is mass spectroscopy can differentiate between these contaminant heavy metals and these heavy metals which I have purified and processed and have given treatment before that? Thank you, sir.
So mass spectrometry is of two types. One is inorganic and other is organic. So you are talking about if inorganic. I have discussed about ICP-MS, inductively coupled plasma mass spectrometry. You can take if you are having some plant and extract it, you can know the composition of elements in that. One plant you can take from Delhi or most polluted city, or you can take from Uttarakhand. Then if the species is same, then there will be difference in the one is growing in the polluted area, contaminated, and other in the clean area. So before going for that species, you need to which plant you are making use of for your medication person. You need to know the source of that. And if you are not knowing, still you can try it if it is in the form of medicine. Even then you can get it testified with the help of ICP-MS instrument at any lab.
But their structure or their amount would be it would be added both.
That will depend upon the climate where it is growing. It will take from the roots.
Yes, sir. Sir,
So-
in when you are doing mass spectroscopy for my medicine, will you be able to tell me that this much mercury is because of medicinal wala and this much mercury is a contaminant? Can you differentiate by mass spectroscopy?
We need to do some control experiment.
Okay.
You need to first take the medicine and you need to take the resource. Do the analysis simultaneously using some standard. You will come to know that whether it is contributing from my vegetation or it is from my...
So separate analysis is needed.
We can do some control experiment.
Okay, sir. Okay. Thank you so much, sir. One more question I have. When we are doing when we are using mass spectroscopy for complex Ayurvedic formulations, which are having animal proteins also or animal fats also, herbs also, heavy metals also, volatile oils also. So will only mass spectroscopy be enough to analyze all the constituents, or we need different things?
Different. If your analyte is volatile, then you need to do the liquid GCMS.
Okay.
And if your second thing, it is stable or not. It should be volatile as well as stable. It should not disintegrate.
Okay.
Then you can make use of GCMS. And if your sample is in the form of liquid and is not volatile, then you can make use of LCMS. But if you are interested in some complex structure, alkaloid, terpenoids, natural products, then you need to go TOF, because the limitation of the detector, their mass detection limit is not so high if we talk about GCMS or LCMS. Depending upon the analyzer, which analyzer for which you need to screen the whole sample for the full range to know the composition of your analyzer.
So we have to provide multiple samples for different.
You can go first you can go through the literature and what are the expected and what you are targeting. And if it is below 1000, then you can make use of GCLC. There is no demarcation between some sample can be analyzed with GCMS as well as LCMS.
Okay, sir.
So you need to do some practice, and after that, going through the literature, you will come to know which is suitable for, or earlier what people have used for this analysis.
Okay, thank you, sir. One more question, sorry. At the end of your presentation, you told about MS imaging, and that is a remarkable thing. I am not aware of that. So, how is it available only at lab level, or it is already out there in the market that we can prescribe to patients?
So, it is some companies, say Shimadzu is from Japan, they are manufacturing it.
Okay, sir.
And though it will be not so cheap, and one problem is the cost effectiveness of the technique.
Yes.
So whether we can afford, so we need to work on this, the cost effectiveness, better sensitivity, better detection limit. So BARC, Bhabha Atomic Research, they are manufacturing mass spectrometers. But they are indigenous, so our most of the purpose can be sold. But even then, so many institutes are procuring from foreign. So we need to
So it
promote
is,
the make in India.
it is yet to come for use
Mass
for
imaging,
us.
we can procure, but that might be somewhat costly.
Okay, thank you so much, sir. Thank you.
Any more questions? Ma'am, please pass the mic.
Thank you for the session, sir. I had a question concerning taking the reading from the data. Like many a times, we'll have many unidentified elements in that. How do we identify those elements? Like libraries are there, but which is the most ideal library to go for?
NIST.
NIST, but still we'll have some pending, no, sir?
Then you can enrich your own library. Some species are not there. Like LC also having one library is also there. I have forgotten the name. But most of the people go for NIST library, and company also sell the list. One more library is there. I forgotten the name, but NIST is most popular.
Yeah, NIST is commonly used. So then what about that detector counts when you say?
Huh?
Till what level? Like I got the new information, it's up till 10,000. But within 10,000, you will have multiple repetitions. And even beyond 10,000, you will have multiple repetitions. So how do you take that? Like you said, isotopic variations will be there.
Then
Mass,
if there are isotope, I have told about iron mass spectrometry. In that, if I have shown the example of paper,
ICPMS.
if you are isotopes, then you can do isotopes, ICPMS. If you want to interested in elements, then ICPMS can
No,
do.
sir, like if you are going for TOF LCMS and we are having the data, and in that within the detector count of 10,000, we are having repetitions.
Repetition, I don't understand what you mean.
One element is coming twice or thrice with
Then their
different
mass will be
detector
different.
counts.
If they are isotope, then their M by Z will be mass.
Slight variations are there in
Then
00.
that depends upon the resolution of the instrument. If the instrument is able to resolve, TOF can resolve that. Up to 4 decimal places it can predict.
Yes. So, but as a researcher, how do we present that data?
So you have to show, I have shown the data of mass.
Rishi, please change the slide. Slide.
That last slide. Back. Back. Next one. There was some calculation on the previous slide, I think.
Previous slide.
Previous slide.
Previous.
This one. So this is the mass accuracy. So this is the difference between the two peaks. 400.00 or 400.004.
Yes.
If what is the difference? 0.004 divide by at which exact mass is this 400, we divide it, multiply by 10,000, it is coming out to be 1 ppm. This is the error, you can say. And if the instrument is able to resolve these two peaks, then it's fine for your measurement. And if you are going beyond then it is not fine. You have to look another option for. What TOF will serve your purpose? You can easily distinguish. You will get two peaks. These peaks will be resolved just like this. If the resolution is less than, if the instrument resolution is 1000, we will not be able to we will not be able to see them, these red peaks separately. But if we go to an instrument which is having resolution of 10,000, then these red peak will be clearly visible and you can distinguish between the two.
Okay. Thank you, sir.
Thank you, sir. Any more questions? Now I request our chairperson for the today's session, Dr. Vedantam Giridhar sir, to give away the remarks for the session.
Sir, be seated, sir. Dr. Bhupender Singh, sir, and Dr. Ramakrishna, sir, and other senior faculty from Sushrutha and other colleges, delegates. It will be my pleasure to be here because the college, Sushrutha College, and SVASA has invited me, and I am indebted to all of you for inviting me. From my heart, I thank you all for inviting me. And here, this session is more scientific and we have more applications. And as one or two faculty who are dealing with such studies have come with questions, because usually we are not doing at PG level, we are doing at PhD level, and some are doing PhD guide. That's why they could get this. Actually, now it is the time we have to move to the level, because our publications, if you go to NAC, many of our colleges, we are having a publications which are below UGC. All are counted as nil only. We need to have publications at UGC and Scopus and Web of Science. Actually, UGC journals also we don't have any value because you don't have impact factor for that, and you don't get H index also. So for that purpose, we need to go to a journal which is either Scopus or Web of Science. For that purpose, we need to have more scientific data to our studies. We are doing empirical clinical trials, RCTs are accepted and we are getting publication. But there are other areas where we have to concentrate. One is whenever we send any article to any what we call indexed journal, highly indexed journal, they say you are using one drug. Is it characterized? So you need to have a characterization as per the present scientific level. If you have with an TLC paper, etc., it is not acceptable nowadays. So you need to, because the science has level, grown to the level. Now we have ourselves question, madam question, that level, 10,000 level also we are able to cross, means our Ayurveda is posing question to the science, you are unable to have a resolution where our Ayurvedic analyte is exceeding that limit. So then science will come, scientists and engineers and many people will come and they will solve the question and you will get a new instrument for Ayurveda. So we need to go to the level of the present science. That's why we have to come with this. And second, we had some discussions when we had in Kelly, Thomas Jefferson University people came and we need to have some integrated protocols, etc. So they told three things are needed. If any drug has to be trialed in US, they need three. One is very sound clinical documentation on the drug, that is efficacy level, and safety toxicity data should be provided. So we can do these studies. Safety toxicity can be done in animal and you can have good RCT and you can have a clinical trial and you can have these two data. But the third point they told is which we are lacking. That is, what is the mode of action? What is the proposed mode of action of our drug? It is a very complex question, because when, as sir told, there is analyte in allopathic medicine is single chemical. Analyte in Ayurvedic medicine is not single analyte. And we have enough information of analyte of allopathic medicine, like our diclofenac or anything if you take, you know its kinetics. What happens in the GI tract, how it changes, how it will be absorbed, how it will be metabolized. Every information is available. and how that molecule, final molecule is attached to the receptor and eliciting the action, and how it will be eliminated. These are all information is available. But for Ayurvedic drugs, because they are complex, you have, if you take a single herb, you are having hundreds of phytochemicals. And when you are giving a polyherbal preparation and herbomineral preparation, you have very big complex material where you have thousands of analytes present. So here comes the problem. Why we are unable to do is, this is the situation. We have multiple chemicals present in the same drug. When we are giving, how we will be analyzing? How you are able to analyze the absorption? So this is a big question. Actually, pharmacokinetics in Ayurveda is not yet solved by the present science. Because this is the challenge to the science actually. Because scientists have to take up the challenge and they have to come with the solution. We are having some of the areas where, like our heavy metals also, characterization, as madam is asking one question, can we differentiate between our purified metal ion with the contaminated metal ion? It is not possible with any science until now, because you are making that into specific ion, you are making that, you make that into Hg, you are making that into ion of Hg, so all the bonds which are made are removed. Then how you will make in ICP-MS? ICP-MS cannot solve that. So you need to have some different, as XRD or some of the things are there. Now these things can solve, as sir told, because as there are two parts, as sir told, there is an inorganic as well as organic. When you go for inorganic, you are going to the elemental level where you are unable to understand what is the process, means what we are making, Shodhana, Marana, or Bhavana, what we are making, we are attaching our herbs, we are making into some colloidal structure, or you are making them, the metal into, trapped into a lattice. That will be missed when you are making AS or ICP-MS. This studies like tandem or something, because they are studying about macromolecule, they may also be able to identify the metal inside also. So they will be coming with a total molecular formula, means molecular weight we will get. So then it is the mass spectrometry scientist will be able to decide what is the molecular formula. So once you get the molecular weight, then they can get, as sir told, libraries there. It is not that much simple. We cannot run with one thing and go then come with your molecular weight. It is not possible. It takes years. It is very big thing. And we should understand why, because as he is showing so many things, there are so many points which are to be kept in mind when we run to a chemist also. So here what is the problem here is, we have to understand the multiplicity or complex molecules are present and how they can be separated is one of the important thing. But some level means you have to make into amsha, means we have to make that into smaller things and we can do study. So one study some studies, one or two, we have done for some, our Erandam Niru Basti. So actually one study was conducted earlier Jamnagar on rats. So they have given the Basti and they want to see the absorption happens or not. Then it was taken for a clinical trial and we want to study the kinetic parameters, whether some of the chemicals present in this Erandamol Niru Basti will be absorbed. Can we see the kinetic pattern of it or not? So we choose the HPLC method. So when we have done HPLC, actually we have done UV. We have taken actually we have done the UV spectrometric analysis of Erandamoola Nirobasti. Then we have given it to rat. Before and after we have collected the blood, we have taken the plasma and we have run. And we could find some peaks which are there in Erandamoola Nirobasti are also present in the blood after giving the Basti. So we thought, okay, some peaks are coming, yes, if we go to high level analysis, we will be able to find out some chemicals. So we want to keep one molecule also, like sir told standard should be there, we kept one ricinin also, and we found ricinin coming when we are doing with UV. And when we have done this HPLC, what happened is, we are getting ricinin at certain timings, but not the same type in all the animals or all human beings. So there is difference, and then we found LOD, what we call limit of detection. So the level of renin what we are getting is below the LOD. So when we discussed with the mass spectrometric analyst, so then he told, no, you are doing with HPLC, its resolution, what you are doing, that it is beyond. That's why you have to go to LCMS minimum. First you do LCMS, then you have to do. So unfortunately what happened is, by the time we gave sample, no, it took around 1 month for our HPLC. So when you have given, when you have done, you could not get anything. So there is, what will happen, no, this is also, renin is also biological material, and there is degradation. So because of that we could not trace anything. But means we need to understand which type of analysis has to be conducted based on the resolution. Now the analysis level has gone to very level, MALDI and TOF, these are there. So if you do HPLC, you are not going to get anything for our biological Ayurvedic products. You need to do the analysis using MALDI, TOF, etc. only, because like the proteins what they are analyzing here, our biological botanical molecules also are complex structures, so they can also be studied through these things. They are also organic molecules. That's why we need to go for such a study where we can come with two things. When you do this MALDI or TOF, etc., you are getting two type of data. One is the metabolites of our plant material. You will be able to trace in the blood where they are there, at what time they are there, even if you go to tissue level also, whether they are reaching the tissues or not. Because as I told, you can use the tissues, you can use the blood, plasma, CSF. So you can use different, and then you will be able to find out these drug metabolites are coming into the tissues, blood or not. That is one thing. Second, we can also see the effect of these drug metabolites on our body. So when they act, then there will be interaction with the receptor, then there will be signal transduction, then there will be then DNA, then transcription, translation. So these all things. So then what type of analysis we can do is, we can do the metabolomics, proteomics. So these are omics studies. So these omics studies, you can understand the body metabolites, and those can be analyzed because they have standards. For plant metabolites, standards are less. For body metabolites, so much work is available. As sir is telling you, there is huge libraries are available. So you can study the effect of our drug. Actually, you may not know what is the exact chemical which is acting, but you can say this metabolite is found, this metabolite is decreased, this metabolite is increased. So then we can say this is the mode of action of our drug. You can say we have given this drug, it may have 10 or 100 metabolites, drug metabolites, but it has changed following metabolite in our body. So through metabolomics or proteomics, you will be able to tell the mode of action of drugs using these mass spectrometric studies. So it is one of the very big thing which if we show this, then definitely everybody will accept. This is called science, because this is the highest science. Nobody can say this is wrong. And one more points are told, that is biomarkers. So this biomarker study is a very big thing actually. So actually we study these biomarkers for each disease. This like HbA1c is there, or many HGOT, HGPT are there. There are so many biomarkers are there at metabolite level also. So these metabolites are nowadays studied, and there is a very big scope for Ayurveda in this area, because today morning we are discussing about gulma. So in allopathic context there is no specific disease for gulma. So we can have a clinically diagnosed gulma cases, make the metabolite analysis, come with biomarker. So we can come with biomarker for Ayurvedic disease names or Ayurvedic disease complexes what are there, we can come with biomarkers. Not only we can come with biomarkers, I have usually, earlier we had discussion with IIT Dharwad sir. So there they are doing some studies on urinary markers, as you told, they are collecting the urine and then they want to do this diagnostic kits, sir what told. So they want to make a diagnostic kit based on urine. They want to early detect the BPH. So the present studies are not making the very early diagnosis, and blood based analysis is there. So they want to come with urine based analysis. They have done some work and they came with some patenting also, and they are coming with some kit. Then there only I identified, you can not only identify biomarker or metabolites which are there in the disease, then you can also make a pathway analysis also. How a pathway is there. So if we have a Ayurvedic disease condition like gulma, and we have a clinical presentation, we have collected what we call the either blood, what we call urine, or body fluids, what we call saliva, etc. Then we will be knowing what are the metabolites present, and we will be able to come with a pathway for the disease. So these are some of the new areas where some of the diseases which are mentioned in Ayurveda, we can come with very good scientific understanding how these diseases can be understood in the modern molecular biology way. This is one thing which we can do. And one more thing is the characterization part. So for characterization also we need to have some more instrumental analysis, and we have to discuss with the scientists, those who are working, so that as some of the problems of resolution, some of the problems of what we think, when I am thinking that it is gold or mercury which is having a different structure, but when we do analysis in ICP-MS or something, I am getting the same thing. So how we can differentiate between these two things? Because we have done one study where we have done an ARCA. So there are around 300-400 ARCAs are mentioned in one book. Means it is a distillation process. You have to put the drug and you distillate and collect it. But in that process, actually, if you see, they are telling that when volatile comes, you have to remove. So you have to collect in the middle part. So many of the drugs are not at all having any volatile material. But still when you make ARCA, they have some organoleptic characters. We have analyzed some of the ARCAs which are not having any volatile compound in it. But when we have done analysis, we have done very simple analysis, sir. We have not done instrumental analysis except UV. We have done physicochemical analysis, we have done UV. And we could get different peaks in UV. Earlier, when we have taken water which we have used, it is plain. When we have made ARCA, you are getting some peaks. Actually, there is no volatile material is found in the literature, but still we are getting something. It means there is something, even non-volatile what we are thinking, there are some volatile material, which might not be found in earlier studies, but this ARCA process is bringing them into the water and which can be given in the treatment. So they will come in very micro level or nano level, so that it is a nano or micro level dilution method of giving the drugs. So there are many of the Ayurvedic areas where we can use the high-end instrumentation like mass spectrometry, MALDI, TAF, and these hyphenated techniques like GCMS or LCMS-MS. So as told, LCMS-MS is also there. So these things will make a more accurate prediction of our structure, and then we will be able to come with how the drug will be there. And one of the very important point what is happening in 2 hours also, we could not find this actually, because we are not having a single analyte. When we give a drug, what is happening to the drug? We thought there is resin. We analyzed the drugs. We know that when you have done HPLC or LCMS or something, you found these are the phytochemicals present. We know these markers. But when we give the drug to the patient or to the animal, and when we want to do the kinetic parameter, what will happen is these chemicals cannot be found. Why? Because absorption changes the molecule, and digestion changes, absorption changes, liver changes it, blood also makes interaction with it. So when it goes into the tissues also there is interaction. So what is the final chemical which is there? That cannot be found. But as you told with MS, that moiety of that molecule can be found out actually. It may be broken, but you may be able to find out that what is the actual molecule, how it is traversing into the body. So that type of analysis also can bring how our molecule is changing through the digestion and metabolism. So there is ample scope of study of high-end mass spectrometric analysis in our Ayurvedic sciences, which can give very good scientific evidence for the mode of action, understanding the proteomic, biomic, what we call metabolomic type of understanding of our Ayurveda, so that worldwide everybody understands how this will happen. We have done one study in a cancer group of patients where already familial cancer is there and virechana is given. And when gene expression is studied, whole genome expression is studied before and after. Actually familial means there is genetically already it will be same. But the expression when virechana is given in a classical form, the expression changed. And they found that BRCA gene is one of the cancer gene. So the expression has been inhibited. So it shows that when we are, this is called as what we call epigenetic type of character. Means genetics are there, we are giving our external things, environment is changing the genetics. It is called epigenetics. So our Shodhana therapy is epigenetic. So it is reducing the BRCA levels expression also. So I thank our Bhupender Singh sir for enlightening us with his very vast knowledge on mass spectrometry and its application to the biology and degenerative disorders. I thank you sir, and I thank the organizers for inviting me. Thank you all for your patient hearing.
Thank you so much, sir, for your remarks on the session. This was the true intersection of innovation, technology and Ayurveda. So I request all the dignitaries to felicitate today's speaker, Dr. Bhupender Singh sir. I request our director, Dr. BRR sir, Vice Principal ma'am, MS ma'am, Ramchandra Bhatt sir, ex-Vice Chancellor S. Vyasa, Agora Murthy sir, today's chief guest for the valedictory function. Please join us on the stage for the felicitation of today's session speaker, Dr. Bhupender Singh sir. I request our principal to join. I also request Giridhar Vedantam sir to join for the felicitation. I request PG Dean Dr. V.V. Reddy sir also to join on the stage. Dr. Vedantam Giridhar sir. Dr. Vedantam Giridhar sir. Thank you all. We have come to the end of the scientific sessions. I request all the delegates, there is stalls on the right side of Sanskriti Bhavan. You please have a visit and be patient. We have a valedictory function in another few minutes. Thank you all.